The projects that come under this heading here, there's actually 3 of them, and we have 3 different environments. So I'll just talk you through each of those different environments and what we kind of learned during that. But overall, this is a study that was funded by NASA through the Human Research Program. It was Gene Hunter, Dr. Gene Hunter at Cornell University is the principal investigator. Apollo and I were recruited as postdocs to work on this. And we have Bruce Halpern and Kim Binstead as co-investigators of this. So even though this was done under the banner of Cornell University, the study environments are actually nowhere near Cornell at all. I was sent down to Texas, to Galveston Island, where there's a Bedrest facility there at UTMB. Apollo did a lot of his work at Cornell, but the actual experiment was done in the skies above Texas. And also we built a facility in Hawaii. Funnily enough, Hawaii actually looks very much like Mars if you actually get into the mountains. It doesn't look like it near the beach, of course. And so we're very careful to tell you that we're not down in Waikiki or Honolulu or anywhere like that. We're on the big island and the lava slopes. And I'll show you some pictures about that as well. It's a pretty weighty title, effects of retronasal smelling, variety and choice on appetite and satiety. Now, retronasal just means smelling through the back of the nose, and I'll explain that to you as well. But essentially, it's a food study, is how we're looking at it. And the reason why we need to study food in space is because a mission to Mars is going to challenge our food technologies. It's a long, long trip, and it has a component of weightlessness on the way there and back, and a component of being on the planetary surface. But the planets that we're going to head to don't have the same gravity as Earth. They're much, much lower. And so how do you cook in 1/3 gravity or 1/6 gravity, say on the moon? So that's what we wanted to also examine. The problem that we're finding in space is that we need our astronauts to eat well. They need adequate nutrition, and they're not. There are problems with getting enough nutrition to our astronauts. Part of it is that previously there was a low variety of foods available for them and no fresh produce. So there was a lot of food boredom that was going on. Gene Hunter's study examines this question, well, how can we eat well in weightlessness? And also, how can we eat well when we're on a planetary surface? So in weightlessness, NASA has a huge variety of foods available for the astronauts, and they're really nice. In fact, they don't really look like the ones that you see around here. As you can see, there's like crackers, there's orangeade, there's peanuts, candy-coated peanuts. That kind of looks like the cream spinach that you've got there. But they have a very broad menu. They choose the menu themselves at the moment at the space station. It's not a prescribed menu. They're not told they have to eat this kind of food on any set day. But they know that whatever they choose is going to be freighted up 6 months in advance. And it's going to live in their locker for that time. Hey, welcome. Come on. Yeah. The problem with the prepackaged food, though, is that there's very little flexibility to actually mix it and create new recipes. So while they have variety at the moment, they don't have the ability to be creative with the food or change the actual ingredients themselves. They can change what they want to eat in a meal, but they can't actually mix it and create new things. So the astronauts, even though they have all this variety, are still complaining that food tastes bland in space. And some of them are taking out Tabasco and chili to add to the food, especially on the space station where they're up there for 6 months at a time. And we wondered if there was a physiological basis for this. The reason why is in weightlessness, your body fluids move into your upper body. Okay, this happens almost straight away. So you get like facial stuffiness and you get nasal stuffiness. It affects your eyesight. It affects your astronauts complain about headaches. And so we wondered if there was a relationship between the food tasting bland and maybe a lack of smell caused by this fluid shift to the upper body. So that we wanted to study that. Once the astronauts land on the planetary surface, you can cook again. So here's the possibility of being able to mix ingredients again and do some cooking. The idea is space farming. We kind of wanted to do that. It's not really part of our technology at the moment, and it's unlikely that that'll be available for the first few missions to Mars. One of the reasons why is that we just haven't developed a technology that can enclose farming within an enclosed environment. The other is it takes a lot of time to farm. And do we want our explorers in the first few missions to Mars spending all their time, just farming food? So it's probably unlikely that that will be there available at the time. So at the moment, NASA's talking about two competing food systems, and what we're examining is both of these. One is continuing the prepackaged food system, and the other is using ingredients like this and mixing them on the planet's surface. So while this is very user-friendly, it's easy to rehydrate, it's quick, maybe we don't really want them to be just using this on the planet's surface. Maybe we want to give them more flexibility and be able to create lovely dishes like this chicken and potato right here. Eating, freeze-dried food that they can mix together. Chicken in there, raspberries. I mean, I can hand these around to you if you like and just have a taste. You can actually just put them straight in your mouth. Drink some water if you like, but you may as well dig into those and see what they. Some packs that you can just hand around and. So we're researching, of these two different systems, which one would seem, the most useful, most flexible, and most likely one to use. And they both have advantages and disadvantages. A prepackaged food system, there's a lot of packaging. There's a lot of recycling, I guess, to be done in terms of that. The other is, if we move into more cooking, we need a lot more water, a lot more power to do this kind of thing. So these were examined in our analogues. So Dr. Hunter proposed 3 years of study to have a look at this. The first is weightlessness. How does it affect the sense of smell? Or does it in fact affect the sense of smell? And is this causing some of the problems we're seeing, say on an outward flight or sitting in the space station where astronauts are talking about food being bland? Two, although food can be cooked, cannot be cooked in weightlessness, it can be cooked on the planet's surface. So, which type of food system should we use? A prepackaged one where it's just rehydrating, or can we actually open up to a much more flexible, creative kind of food environment? And how much resources and labor is that going to take? And the third is something that Apollo will talk about, sauteing, frying, boiling in low gravity. It really hasn't really been done before. Well, actually, we just did it recently. But it hasn't been done on a planetary surface. How does all that spatter and cooking, how is it affected by low gravity? What kind of cooktops would you need? What kind of galley would you need to do? And that's something that we're looking at engineering. So we involved ourselves in three environments. I was involved in the Bedrest study. This is where we put people in bed for 70 days. Just in case you can't, so my accent is 7-0, not 17 or 7. 70 days with their head lower than their ankles. And this simulates the weightlessness that we see in space. So they feel the body fluids moving into their upper body. I tried it myself for 4 hours. I didn't like it. I got back up. Okay. I felt I needed some empathy with them, so I could work with them. But they stick it out for 70 days, and they cannot get up at all for anything. Okay. So they have to use the bedpan. They shower in this position. They can roll on their side and on their front and stuff, but they just cannot. The only time they're allowed to get up is when they eat, and they can prop themselves up on their elbow so that we don't get them choking when they're eating food. Okay. So we had 16 subjects enrolled in this, and we were doing a study on food acceptability and smell. And this was done at the Flight Analog Research Unit down in Galveston, Texas. There's a facility set up there. It can take up to 10 subjects at a time. We were often running 2 or 3 at a time while we were doing that. To study preparation of food on a planetary surface and to look at these two competing food systems, the prepackaged and the shelf-stable items that could be cooked, we actually built a habitat on the slopes of Mauna Loa on the Big Island. This houses a crew of 6. And they are isolated in this for 4 months. There's a 20-second communication delay, so it simulates being on Mars. And during this study, They were asked to cook, just rehydrate the food for 2 days, and then the other 2 days they could cook using any of the ingredients on the shelf stable. There was no fresh produce, no fresh milk, no fresh dairy. It was all freeze-dried food. Okay. And lastly, Apollo and I got the opportunity to go on these parabolic flights to test out Apollo's design for this new galley that could work on the lunar surface. Apollo's got a great video showing you how all that works, but it was a very fun activity. We got to experience weightlessness many, many times, and we got to cook successfully in a lunar gravity and in a Martian gravity. So I'm going to talk to you about the Bedrest facility now and some of the stuff that came out of the high seas, and Apollo will then follow talking about the zero-G experience. So in Bedrest, we were doing physiological measurements. We were measuring nasal tissue swelling or nasal congestion. So as these fluids moved into the upper body, nasal tissues would swell, reducing the volume of the nasal cavity. And we could measure that using an acoustic rhinometer and also measure airflow with that. We had 2 groups of subjects from our 16. 8 were low exercise subjects. They did very little exercise. And the other group were exercisers who performed a lot of the exercises that are being performed on the space station because they were testing that. So that was a really great way of having a look at some of our effects. We also measured their smell using a whole battery of tests. We measured their response to smelling the food from the study that was served to them in the study, also smells that we brought into the study, and also standard commercial smell tests, which some of you can try out. I've set up some on the back table there, and we can see how you relate to some of those kind of smells that we were using. One of the findings, probably our main finding, now, a lot of this data we're still kind of working on it. Our last subject finished in June, so we've got a lot of data that we're still working through. So this is a profile of the nasal volume changes, okay? So this is tracking nasal tissue swelling through the study. So this starts at day 1 of bed rest, okay, here. And this yellow line here is baseline. This is the average baseline nasal volume. So we measured them 7 times before they went head down. Then we were able to measure them every 2 hours during the first day. So we got 5 measurements from them then. Every 3 hours on the second day, and then daily up to day 5. And then we measured them weekly all the way through the 70 days. And then we measured them daily when they first came back out of bed rest. And these are pooled values. They're a percentage difference from baseline. Okay, so this is the amount of change that occurs from this line here. So as soon, like within an hour, we measure them in the first hour after going head down. We see this dramatic reduction in nasal volume. Okay, and it's the same for both exerciser and non-exercisers. They all show, an effect of going head down, and this is what astronauts report to us when they're in space. And then that kind of fluctuates throughout the day, but it generally stays low. But within 24 hours, they're starting to adapt. So on the second day, we see that starting to move back towards baseline. And then over the next few days, the body is still kind of adapting, and there's a whole lot of different physiological processes going on. And so we see that returning again to a more stuffiness in the nose. What was interesting here for us is that we weren't expecting that there was going to be a difference between an exerciser and a non-exerciser. And what we found is that the exercisers, nasal tissue swelling was much, much less throughout the bed rest study, and almost returned to baseline by the end of it. Whereas our controls seemed to be affected more by the fluid shift to the head and remained stuffy, basically, throughout the study in that sense there. Except for this one weird little blip here, which we're still trying to work out why all our controls showed this almost return to baseline halfway through the study. Maybe they suddenly got happy they were halfway. We're still trying to work out exactly what went on in that because it's the same for all our controls. So it's not just a couple of our subjects pushing the acting as outliers. This is really almost clinically relevant too. This is not just an interesting insight for space. This is telling us that for our patients in hospitals or anyone who's like bedridden for a long period of time, a little bit of exercise, or maybe a lot of exercise, is going to help with their bed rest. and certainly in terms of like nasal stuffiness, we know, this is what it's telling us, but we expect that in the head, that with all the organs, the eyes, the ears, that, a little bit of exercise is going to certainly help with being bedridden for long periods of time on Earth.
How do you get exercise in bed?
Oh, it's great. What they do is they have them lying down, right? And they put them in this harness, and then they pull the gurney out from underneath them, and then they actually have a vertical treadmill, and they actually run up the wall. So they jog. They have especially adapted leg press, so they can lie down and do resistance exercise. And they also have a power cycle too that's on its side, so they can cycle while they're lying down. Because we don't want them to sit up, because a couple of hours of them sitting up doing exercise is probably going to mitigate any effect of the bed rest probably on that day. So yeah, they're fantastic, and a lot of those videos are available through the NASA website. They're really great to watch. It's kind of cool. So it doesn't look like there's a huge effect, particularly of volume. I mean, it seems like a small effect, but it's airflow that we're most interested in. How would airflow affect smellability? And so if we look at the actual airflow that's produced, again, we see a huge difference between controls doing low exercise and exercise. In fact, exercise almost look like their airflow is better through their nose during bed rest. And certainly in terms of the return, they seem to have a much better airflow during that period. Whereas controls, again, still seem to have, bed rest still seems to maintain a low airflow for them. And one interesting aspect about this here is that once on the first day out of bed, of course, once they get up, all their fluids drop away from their head. And so, of course, the nasal passages open right up, and we see this big bump here. And then eventually it comes back to normal over those 2 weeks. That is the only bit of data that we can compare directly with astronauts, because we know when they come back from space, through different measures they've made of the pressure in the eye, that the pressure is dramatically lowered. So we know that this is very similar to what astronauts experience. We would like to get this equipment on the space station and actually test them in space, because we suspect that the space environment is even more harsher in terms of fluid flow to the head. And we probably expect to see a bigger effect in this kind of thing. Another thing that we wanted to sort of compare with astronauts, and this is kind of showing how relevant it is in terms of that what we were doing in bed in astronauts, is that out of 664 astronauts that have been in space, 60% of them complained about nasal congestion. Okay, this is in the NASA medical library. Let me tell you this. Okay, out of our 16 pillonauts, 56% were able to self-assess themselves as being congested, because we asked them that question before we did our test, to rate their nasal congestion at that time. But we know that all our pillonauts suffered nasal congestion. So we suspect that probably a lot more of the astronauts were suffering nasal congestion, but just didn't kind of realize it, or just didn't feel like telling anybody about it. So we think this is a much bigger problem in space, now that we know exactly what the fluid shift is doing to the nose.
How does dehydration affect the fluid shift in space versus,
Yeah, it has a whole lot of different effects. And also, there's a whole lot of fluid intake and fluid uptake and plasma volume loss that goes on. So some of that is mitigating some of that effect in the nostril. But astronauts are very aware, and our bed rest subjects too are very aware, they had to keep drinking. They have to, because they're excreting all the time in those first few days. The heart sees this amount of sudden volume there, and it's going, wow, you've just drunk 5 pints or something. So get rid of it, So they have to keep drinking until their body gets back to that, to an equilibrium or homeostasis, with that. So we try to make sure that doesn't happen. Dehydration is a very real kind of aspect. But yeah, if you couple that with, that fluid loss, it's definitely going to have a big effect on the physiology and stuff. There are a lot of processes in the body that slow down during that weightlessness as well, because of that shift of fluid and the way the body senses that and reacts, accordingly to that. Okay, so we also tested them for smell, and this is kind of a fun thing. They like doing this. We had every time we did the nasal patency measurements, we also got them to smell 20 different smells. Blinded, they couldn't tell what they were. They didn't know what they were until the end of the study. We got them to smell through the front of the nose, okay, which is kind of normal. It's like the way you're smelling now, just breathing stuff in. But while you're eating, with your mouth closed, with good manners, the odours actually go through the back of your nose. And that's how the sense of smell and taste are connected as you eat. So you can smell your food to make sure it's fine and then you're not going to eat anything really horrible. But then when you're eating, the smell drifts up through the back of your nose and you get that sensation of smell and taste interacting with each other. That's called retronasal smelling. And so we tested the orthonasal, which is from the front of the nose, and the retronasal from the back of the nose at the same time in a randomly selected bunch of smells. And we took smells from the menu so we could test whether there was any change in the way they could smell foods. And their menu, like applesauce, mac and cheese, bacon, potato chips, all those kind of things. Off menu, we kind of brought a few more spicier things in, like garlic, chipotle, parmesan, some softer smells like raspberry and root beer. We were a little bit mean because we actually made them smell coffee and chocolate, and they weren't allowed to eat those during the study. And I tell you, I was having to grab that coffee container off them at sometimes because they were just huffing on it, once they realized what they were smelling. And then we had a few calibrates, which are, really like banana, coconut flowers, peppermint, and soap, which are what we know are very strong and usually the ones that don't change for most people. And we found that in 10 of our subjects, there was a decrease in smellability. And this is a self-assessed, self-assessed through them rating their intensity of the smell over time. So in this portion here of the graph, this is their initial ratings on a scale of 1 to 5 of what they, of how intense they felt the smell. Coming through. Come on in. And then you can see in this particular subject, in Bedrest, not a big change on the first few days, okay? So we're not really seeing that, the initial fluid shift to the head is actually causing a smell decrement. But as you can see over time during the Bedrest study of the 70 days, this ability to smell is slowly dropping off. And in most cases, it didn't really return at the end, okay? And that's from the front of the nose. This is from the back of the nose. The green line is the calibrates, so we were expecting those to be pretty flat because they could smell them well, but those also dropped a little bit. Off-menu smells pretty much followed the menu smells. But in all our subjects, it was highly individualized. For some of our subjects, it was only the menu smells that dropped off, and the other smells remained flat. In some of our other subjects, it was the opposite. For some of our subjects, the retronasal smell dropped away, but the orthonasal didn't. So we've got a really complicated data set that we haven't kind of finished trying to piece together. But we have found that, that we had 10 of them showed a decrease of some form, 2 showed no change, and 4 actually showed an increase in smell intensity, self-assessed smell intensity. So, hello.
How do you smell through the back of your nose?
I'm going to show you exactly how to do that. I can actually probably show you now. I'll demonstrate, okay. No, because I had them do it at one cent. So, okay, smelling through the back of the nose. You get a straw into a container, right? You pinch your nose because you don't want to smell through the front. You suck through the straw. That's it. So it takes a little bit of teaching to do it, but it's basically you basically suck it through the mouth and then you blow out through your nose with your mouth closed, and that kind of does that. And we can do some tests so that you can try. In fact, what I like to do is those who are up for it, we'll get you just to smell those smells on the table through the back of the nose, and then we'll see if you can work out what it is, and then you can try them from the front and see if it kind of matches. Okay. So in that sense, so that's.
How many subjects were there? 16.
16.
And
yeah.
You said 10 out of.
Yeah, 10 out of 16 showed some form. Yeah. So I mean that's a pretty good number of them that did that. But again, highly individualized, not completely losing sense of smell, and certainly not frontal back was kind of different for a lot of them as well. So we've got more work to do on that. What we did find though is that only 3 subjects showed a little bit of correlation with reduced airflow or change in airflow. So we didn't really feel at this point that the nasal patency that we were measuring in the airflow is the only factor in terms of that smell change. But certainly Bedrest itself was producing a smell change as far as we could tell. And we didn't find any difference between exercises and non-exercises in this case. Okay, so we didn't see any effect of exercise mitigating smellability or not. It seemed to be very individualized across the group. So the other important thing, and this is something that we can relate to the high seas data as well, is menu fatigue. Okay, this is what I was talking about at the beginning. Things start tasting bland, food starts getting boring. I mean, we know about all this too in terms of Earth exploration because we've seen it, in the Arctic exploration and anywhere where you have a low variety of food choice or no fresh produce or a low frequency of change in a diet. We see this kind of menu fatigue or menu boredom coming up. So we were lucky at the Faro because they give them a prescribed menu. It's very similar to the space station. Well, actually, it's more similar to the space shuttle, actually. It was based on the space shuttle menu. So every 11 days it repeats. Okay, so you think, 11 days, that's enough for me to forget the first time I had that meal. Okay. Breakfast, lunch, and dinner. Pretty standard kind of food, okay? So like the breakfast here, scrambled eggs, sausage, waffle, syrup, strawberries, milk, orange juice, all fresh. Nothing, we're not trying to recreate a reconstituted kind of food or anything like that. And they were asked to, and so because they were getting every 11 days, they got the same meal served to them probably about 9 times during the study. So it's a great data set for us because we can track that. So they got it at least once before they went head down, and once when they came head up, and then just throughout the study, every 11 days. And we asked them to rate, before they started eating, we wanted them to rate every meal item, so scrambled egg, sausage, each one individually. They had to look at it, they had to smell it, and then they had to assess whether they wanted to eat it. Okay, and they rated on a scale of 1 to 9, so the appearance and acceptability, whether they want to eat it. 1 was a dislike extremely, 9 was a like extremely, 5 is, yeah, I could do without that. Interested in consuming the food, same scale, 1 to 9, just slightly different words because you're asking them about that. And we also asked them to rate whether they were hungry beforehand and whether they were hungry afterwards in terms of that. Okay, so what I've done here is I've just averaged the ratings for one meal, not the meal items in this case. Okay, so on this side here, it's just the actual entire meal, the average rating of that meal. So pre-bed rest, we get 1 serving of the meal, and we see in this particular case, this subject likes all 11 meals pretty well. They're up there around about 7 or 8, which is like a lot kind of meal. Immediately after bed rest, not really much change. By the end of bed rest, this subject is hating all the food. And it's all the food, it's all the meal items. And this is what we're calling general fatigue. It's where the entire meal is not liked at all. It's not any specific meal item. Okay. And there's a little bit of recovery post-bed rest, but by then they're just like, I just want to get out of here, and I don't care about the food. I just want to eat it. Same for breakfast, lunch, and dinner. In fact, in this case here, they disliked something on the lunch so badly that it was plunged down to 1, which is kind of rare for us.
What was that? Do you remember?
I think it was the tuna casserole. Fish is always a problem when you have because some people just don't like fish at all. So, so what we see here is this is like looking at individual meal items here, and this is the I've kind of mixed the appearance, aroma, and interest in consuming data on top of the acceptability data, but essentially they track each other quite well. So here we see mixed vegetables, like them quite a lot to start, and then round about day 24 of bed rest, we start seeing this drop coming in. Yogurt, again, round about day 20, we start seeing a drop, and they really just didn't like yogurt by the end. Even though they show really high ratings for it pre-bed rest. And again, carrots, they just really didn't like the carrots. So a lot of the vegetables, a lot of the yogurts, and in fact, actually pretty much all the food really. Dinner fared better than breakfast or lunch in this kind of sense. So this is what we've decided to call it general fatigue, which is, the whole meal system is just broken down for this subject. In this case, doing the same kind of thing of averaging all the meal items so we get an average rating for the meal, it looks pretty much flat. This subject, there's a strange thing here where they're off, but basically it looks like this subject is pretty much liking the meal from beginning to end. There's no change. But then when we actually start looking at individual meal items, we get a very interesting picture going on here. Now this subject came to us and we asked them, what are your favorite foods? What do you like? What you don't like? And he said, I'm all into vegetables. My body's a temple. Vegetables. I don't eat sweet things. I don't like sweet things. So Comfort foods, great. No change at all. Yeah. Vegetables, we start seeing a decline during the study in vegetables, and this happens to all vegetables. Funnily enough, sweet things, which you didn't like at the beginning, loves them during the study. So this is another whole aspect of menu fatigue or menu boredom, where some meal items start becoming liked a lot, and others drop down in terms of their perception as well. So we've got a lot of data like this. Overall, we found that 7 of our subjects suffered general menu fatigue. 2 of our subjects showed no menu fatigue at all. And the rest of them showed this kind of meal item specific kind of fatigue. There were meal items on the menu which they just disliked, but other meal items they were fine with. In that sense. And we looked at the onset of that because we were told that this would happen midway through the Bedrest study. We find in most cases that happen really early on, particularly amongst the general menu fatigue subjects. Although for some of them it happened in the last week of the study. So by the time they got to the last week, they were just suddenly like, I'm just sick of the food now. I don't want to be over. Again, no difference really between exercises and controls. Okay, so there's no exercise doesn't seem to mitigate that at all. It's just a general thing. Okay, so that's the Bedrest study. I'm sorry, there's quite a lot of data that handing you over there, but now I want to talk about the high C study. Now this is mainly a food study. So again, it's mainly about menu fatigue, and it's a much more complicated data set because we don't have a prescribed menu. Okay, the subjects could choose the food, and what I'll do is I'll let Jean actually talk a little bit about high Cs for you. She's on this video here.
Hello, I'd like to introduce you to a project called High Cs. This NASA-funded project is designed to test food preparation strategies for long-term space exploration. To find the best ways for long-haul astronauts to eat, researchers at Cornell University and the University of Hawaii have designed a food study to take place within a 4-month simulated Mars mission. The researchers selected a crew of 6 highly qualified individuals with various scientific backgrounds to participate. The crew will live and work as astronauts in a simulated Mars habitat, conducting their own personal research as well as participating in the food study. In essence, the High Cs project will compare 2 types of food and food preparation strategies. The first approach is similar to the ones astronauts on the space station use right now, eating pre-prepared, just add water and heat meals. These meals include oatmeal, soups, chili, and beef stroganoff, just for example. Pre-prepared meals like these are easy to make, eat, and clean up. The second approach is similar to traditional earthbound cooking. It's the way explorers used to cook and eat back in the 19th century. Since astronauts on Mars would have gravity, they will be able to mix shelf-stable ingredients and cook from recipes. They could even make bread, egg dishes, and sushi. Meals like these are more complex than pre-prepared foods. They require more equipment, time and energy to prepare, and more water for cleanup. But they could help fight food boredom, which leads to astronauts eating fewer calories over time and possibly becoming malnourished. It's possible that astronauts or our crew could favor one approach over another, or they could favor a mixture of food preparation strategies. It's also possible that one approach to food, or a combination of strategies, might lead to higher morale, improved productivity, better health, and more crew cohesion. The High Cs study aims to find out.
So how's it tasting? Is it? You live off that for 3 years on Mars? Yeah. If you had to. I have to say, I don't think we've got too much of a problem on the first mission to Mars, because I think the excitement and the historical aspect of it, I think anyone would eat anything just to be able to get there, right, and come back. But I think as the more we go there or the longer we stay there, then this becomes a problem, of how to adequately feed astronauts and give them nutrition initially when you can't do space farming, and then later when space farming comes into play. I mean, how much time do you want to spend, creating fresh produce and things like that? The other thing too is the return journey is going to be problematic, because after the excitement of being on Mars, getting back into your spacecraft and spending another 6 months coming back, there's psychological aspects too there that need to be explored in terms of how the astronauts will feel psychologically, and whether food will play a factor in helping them be in a good state of mind on their way back to Earth. So Jean was keen to look at these, examine these hypotheses, and these are the hypotheses she came up with in terms of using a prepackaged food system for 2 days, and then where you could only rehydrate, you could only heat, or you could only cool the food, and that was all. Or 2 days of crew cooking, where recipes could be created, water was able to be used freely to make food, and there was a lot of flexibility in how you might manage the different recipes there. Essentially, we're looking at here again at menu fatigue. So the hypothesis is that the crew will experience menu fatigue when using the pre-prepared and instant foods, and that their ratings for an intake would diminish over time, and they will not experience that when they do the crew-cooked foods. And the crew will eat more on the cooking days, crew mood will be more positive, and resource utilization, of course, will increase on the cooking days, and that's something that has to be managed in terms of if we want to go with that system. And what she found is that the ratings were very similar between cooking and pre-prepared cooking, crew cooking days and pre-prepared cooking days. In fact, they were no real changes, although there was a higher variability among the women who ate on the cooking days. Okay, but essentially no menu fatigue as such. In terms of food liking, there was a narrow response of liking, so more likability on the crew cooking days than there were with the prepared packaged food. But again, not a big change here. So we weren't seeing a huge decrement in food liking throughout the whole mission, compared to what we saw with the Bedrest, where we saw major changes in food liking in that sense. And in terms of looking over time as well, it wasn't that at the beginning of the mission they liked it more, in the end they didn't. There was really no change at all. So in some ways, this kind of food system seems to actually aid in terms of menu fatigue. And this was over 4 months, and they're isolated as well. So obviously a certain amount of autonomy is important in terms of food liking, and also the variety as well, obviously coming into play in this case as well, compared to the Bedrest study, where they had no choice of the food. They had to eat it all that was served to them, and it rotated every 11 days in that way. One of the interesting factors is that food intake was increased on the crew cooking days, so when they could choose their own food and recipe, but only in the women. Okay, the men were pretty flat all the way through. So I'll just finish up there. We've obviously got a lot of data, and happy to talk more about that and have questions. And as data comes out, we'll be able to drill deeper into this idea of what's going on here. But just in summary, I mean, menu fatigue is common in the weightlessness analog, but not in the surface exploration analog. Okay, and there's very little difference between meal liking between prepackaged and crew cooking days in high seas. Food intake was slightly increased in the women on cooking days, and during Bedrest, subjects enrolled in the exercise program, similar to the same exercise they use in the ISS, showed less long-term effect of the upper body fluid shifts, and that's a very clinically relevant idea. We also saw a decrement in smellability because of Bedrest. That's the first time that's been shown. In previous studies that have done that, there has been no change recorded in smellability. So this is a new finding as far as we're concerned. And certainly the 70-day aspect, which is about half a mission length for the International Space Station, seems to be having a factor in terms of what we see in terms of menu fatigue and smell changes, which were probably not picked up in earlier studies who use a much shorter time frame for this kind of work. So thank you very much. I'm going to pass you over to Apollo.
Okay, thank you, Bryan, Sil. So, what time is it? Good afternoon, everyone. So, that was a good presentation by Bryan. So we can see that food is really an important part of any space mission. If we're going to stay for a long time on either the moon or Mars, then nutrition and food is a big part of it. And so Bryan talked about the food aspect. I'm going to talk more about the cooking part, the actual design that we need in order to be able to do some cooking in a space habitat. So first of all, the cooking that I'm going to be talking about would be cooking with gravity. So we are not tackling the problem of cooking without gravity because everything will fly off. I mean, the only way you can cook without gravity is you put artificial gravity. Maybe you make something moving, like a moving sort of barrel with everything being cooked on the sides. But if you're in a space habitat on the moon or Mars, you have gravity, so you can cook there. I mean, things will fly off, but they will fly off not as much as in microgravity. So start with this. So these are a lot of concepts have been talked about on what a Mars habitat or a moon habitat will look like. But we're thinking that in the end, if we're ever going to stay for a long time in moon or Mars, there's going to be some sort of cooking involved. Okay? Especially when we start doing the planting, I mean the bioregenerative stuff. So you have to cook your food. Okay, so the challenges would be if you want to cook in it's a closed habitat. So it's a closed habitat. So it's like, and gravity would be different on moon and Mars. So moon is 1/6 of Earth and Mars is 1/3. So this was the high sea. So this is not the actual space habitat. But you can imagine what the challenges are. If you're cooking in a closed habitat, it's like cooking. I know we've experienced like during our college years, we live in a small room, and you cook something there. I mean, everything goes everywhere, okay? The good thing about cooking on Earth is if things go bad, what can you do? Just open a window, okay? And everything will be, you get out of the bad air. But in a closed habitat, whatever stuff that comes out of the cooking will stay there unless there's good provisions to get rid of it. So the challenges that we put in here, the first one is contamination. It's the safety. So it's always the safety first. So you can contaminate your habitat. So cooking produces a lot of stuff that can contaminate your environment, like the gases, water vapor, particulates, and oil droplets. When you do frying, I'm sure everyone sees all the oil covering everything. And there's also the part that, since it's a closed environment, the cooking will add heat to your environment. So if you do it more and more, it can get hot. So you have to add provision to get rid of that heat. And right now, most of the habitat designs would want low pressure. Okay, I think it's more easier for the astronauts to work under low pressure. And problems with low pressure would be problems with boiling. I mean, if you have cooking processes that involve boiling, at low pressure, it's like cooking on top of a mountain. It will boil at a lower temperature, and your food will cook a lot longer than happens on sea level, at normal Earth pressure. And that's also one thing. There's less convection. If there's less gravity, that's one of the things. When you see boiling, the bubbles go up, and the liquid rotates. It steers everything up. But less gravity, less of that thing. So less steering, less heat transfer to your food. So these are possible problems. Now, in the project that I'm going to describe, we sort of tackled this ones first. Like, we sort of thought about what would be the most stressful cooking thing that can be done, and that's stir frying. I mean, it's easy. I mean, you just put the food in hot oil, but then there's the hot oil, there's the gases, and there's the oil spatter. It's going to happen. So that's the one thing that we tackled first. We're thinking that if we can sort of find a solution for that, then the others would not be that bad. Okay. Oops. Okay, so so the idea for us is that we isolate the cooking in a chamber. So it's like a hood. So you isolate the cooking in the chamber. We call it, it's similar to a chemical fume hood. Basically, this is the prototype. So this is the first time that we have tried to study this kind of cooking in space habitat. So we do not know any basic data. We don't actually know how does the oil, I mean, we've done it. When you're doing, you heat the oil and then you put the food, the oil spatters out. Is there a mathematical model that's going to describe the spattering? So that's one thing that we want to see first. Will there be a model? And because if there's a model, we can put it in a computational model and design any type that we want. So this is a very basic. This is mostly for data gathering. And then we made the mock-up. Basically, it's transparent, so you can see how the flow happens inside. So this is the actual one. It's going to fly. This is for our flow pattern experiments. You will see later how it looks. Okay. So this is the first thing. It's been the way computational modeling has developed. You can do flow modeling. So we have a fan there, and you can see how airflow happens. So it can be done using CFD software. So that's part of the study. We try to see how the flow pattern changes if you put stuff in there inside. So this is good because we can do the model in the computer, and we have the actual experimental setup. So we have validated this. Okay, so this is easy. Once you put your model, you can do a lot of stuff in it. But it's missing the droplets because we have no way yet of knowing how the droplets behave. I mean, the oil droplets behave. We know how the air behaves. And since most of the gases and water vapor and particulates, if they're small enough, will follow the velocity pattern, so we're sort of very confident that we can model them together with the flow. But the oil droplets, no, we need data for that. And how do we get data for that? The only way we can get data for that is if we actually fry. We did the next best thing, which we're going to see later. So this is a lot more of like we put stuff in there and see how it changes the flow. You can see that if you put stuff in there, the flow pattern changes. So we can do that. We can sort of predict how the flow changes inside. Okay. And so we had to devise an experiment. So this is the oil spatter experiment. We had to think of a way that we can get data for the spatter and then try to model it mathematically. So this was what we came up. We have pieces of photo paper there, okay? And then we tried different foods, so hash browns, tofu, rehydrated onions. So we wanted starch, protein, and some onions, okay? And we use different kind of oil. So what happens is these are small enough so that we can weigh each of them. You can see the numbered squares. So you can get mass data. And when this spatters, you can see this, red dots. So it looks like, I don't know, something out of CSI, but so these are the oil patterns. So we took pictures of it, and then fortunately there's software, ImageJ. So this is software from the National Institute of Health. It's free software, actually. They're very nice. You can download it. And because the problem for us now is we have this spatter. We're interested in how many there are and what's the size distribution, okay? And also how far away they come away from the pan, okay? So bulk method would be to count them one by one, but that's impossible. So what you have is the software. It's able to identify the spots, and it can measure, tell you how big it is. So in the end, you have data, number of spots, how big they are, and how far away they are from the frying pan. But still, you have to take pictures of each and every one of them. So that takes a while. So this is Earth. Okay, this is Earth. So we did it on our lab. And then the data, this surprised me because the data out of all that spots, you see a pattern. Okay? This is the diameter distribution. There's a pattern. Okay? So these are the numbers of every square. So there is a pattern you can see. And this pattern can be made into an equation. So that was new for us, because we now are able to have an equation that describes the oil spatter. And you can put it in your computer, for the model. So this is for Earth. So this sort of got us a bit excited, because now there is something we can put in our computer and use to design more different designs for our cooking equipment. Okay, so there's more. So it fits. So the different types of food have their own pattern. So the pattern is good. And there's also mass distribution pattern. So we got to see the area and the mass. And so we also analyzed the flow. So we were able to see that the way we designed the galley, if you do some cooking, it does remove the vapor. Okay, for this one, for our design. Okay, so that's why we have the transparent one. We can see how the flow moves. And this is the last part. We have the Earth model. Now we want to see the Moon or Mars model. So like I said, we need this. We want to see how the spatter changes if gravity changes. So we were able to get time on from NASA Reduced Gravity Office. We got to ride the zero-G plane. The G-Force One. It's actually a private company now. It's no longer the Vomit Comet. It's NASA just buys time on it during the Reduced Gravity Office. So we are now able to build something. So this is just a computer model, but we built something that actually looks like it. And I wonder if you're familiar with the reduced gravity plane. It goes through a parabolic flight. Basically, it's like being in a roller coaster. You go up and down, up and down, up and down. And there are portions there where, depending on the angle of the plane as it goes down, they can simulate Moon, Mars, and zero gravity. Okay? I think it's made famous when movie stars do their space movies. Like, I think Gravity, they did it. I think that was the first one that the director wanted to do it, but it cannot be maintained. It only takes 20 seconds for that weightlessness to be maintained. So what happens is you go up 20 seconds, you go down, go up again. So it's 20 seconds intermittently. They do it like 50 times. So, it's good for us. You will see that they close all the windows, so you don't see the up and down. But yeah, you admire the pilots, because there's the one who see the up and down. Yeah. But for us, no, we don't see it. Everything is closed. So, which is a good thing, because even if you don't see it, you feel it. So you sort of like feel your food going up, and then then someone comes beside you and give you a bag. Yeah, they don't want you throwing up in the plane. But okay, so yeah, this is new activity. We just did it in April. So most of the data from this flight have not been analyzed yet. Okay. So here's Bryan, here's me, and we work with McKell Engineering. It's Susanna. So this is NASA. So they do this. So they pick. There's more than one team. So all of these are teams from different schools, and they do their own experiments. Okay. So here's more pictures of us. So this is the one we built, the actual one that we put on the plane, like this one. Okay. So it's very nice, very official. You get to wear a flight suit, just nice, and get to get your picture beside the plane. Okay. And okay, so here, so it's basically what I showed you before, but now we do it inside the galley. So we have again the sheets. We dye the oil red. We use hash browns and tofu again, so to compare with what we did on Earth. Okay, so now it's inside the plane now. So we're doing this inside the plane. So it has to be contained. So they don't want things flying away. And we have put a filter on top. As you can see later, there's strong filters on top that make sure that the odors don't go out. Because one of the things that, because it's the first time that they've done cooking in this plane. And one of the things that they were afraid of is that when there are a lot of crew members around, if they smell something burning, usually on a plane, that's bad. So they might pull the plug so we had to like do a rundown and show them so that they can distinguish the smell of our cooking and stop actually burning on the plane. So that's one. And the other one was that they were concerned about what happens if there's, the flash points. Usually there are certain limits, flash points, temperature, but if it works only on Earth, what happens if gravity changes? It might change too. So we did some calculations and showed that even if all the oil, so we don't use a lot of oil, even if all of it burns up, it'll be contained. There's not enough energy to burn down the plane. Okay, so it's going to be contained there. So it took a while to convince them, but in the end, we flew. So let me show the video. Okay, quickly. Okay, oops. Excuse me. Okay. So we have a 4-minute video of the Aqua Cooking. So this is just to show you that we are actually inside the plane. Yes? So this is us setting up. So we set up while the plane is not yet on the parabola. Okay? So they let us set up. So we're flying. So we have to secure the pan so that it won't fly off. Here with springs. And all those paper sheets are attached with magnets. So there are magnets there underneath the paper sheets. And then I have to put the oil using a syringe.
Stay over here. As soon as you start, you'll see him put the food in. Try to catch that.
Okay, now we're heating the oil.
Release.
So that's when put the food in. Okay, so this is hash browns. Sign that. Okay, so we close it. So this is now moon gravity or Mars gravity, depends on the flight.
All right, coming out. Release.
Okay, so we have to be very coordinated. There's 3 of us. Like there's Bryan at he's the one that takes care of the data. They made it do it twice. Yeah, one on the tarmac and one on the plane before it flew. So here's the tofu. So it's but like I said, there's a 20-second window. If you miss that, then it's not good. So the flight is wasted for that particular parabola. So we heat again the oil, and then when they say release, that means gravity is what it's supposed to be, like moon or Mars. And here's the tofu. Yeah, tofu sizzles more. There's a lot more water in tofu, so it's a lot more spatter.
Coming out.
You can see you have to secure the pan when it's done. So we built a special box to sort of house the pan and the hot food inside it. And it has to be stowed away. There's Bryan with hash browns. And so we have at least 2 trials per. We wanted more, but I mean 4 days of flying, sort of. That's the usual time they give you. Okay, so there's a few more slides. So I'm going to just show you a few of the results that we got. Like I said, it was April, so we haven't really gone through all of them, but we have some results. Like for one, the number of particles. So we counted the number of particles on the proto paper, and we found that moon almost double. So there's more spots on the moon gravity, which a little counterintuitive for us, because first of all, when we started this, we were wondering whether we'll get something on moon, because if you remember, moon, the particles would fly out farther, and we have limited space for them to come down. But it seems that we are not sure. We are still going into the physics of it, because we know how, I mean, the spatter pattern, but we don't know why yet. It could be that there are more bubble formation in reduced gravity, so you have more oil spatter, but we are not sure. So we're going to dig deeper into that. But this is the data. We're getting more on the moon than Mars and Earth once. So there's a slight increase in Mars, but not much. So Mars is 1/3, moon is 1/6 of Earth. And here's some particle size distribution. So this one's something that's, we expected this. So let me just go through this. The green one is Earth. So this is number of spots versus diameter. So this is diameter, okay? You got bigger. So we see that for the Earth ones, the spots sort of end about 2 millimeters. So you don't get anything bigger than that. But for the reduced gravity ones, about 90% is greater than 2 millimeters. So they still go farther. So you get bigger drops, okay, the lower the gravity. So which is expected to, because the escape velocity for the drops, the bigger ones can go farther away. They can move away. So this is also, this shows it in a different way, the color one. So this one is, the blue ones is above 2 millimeters. So you can see that there's more for the lower gravities. So that's one of the things we saw. So particle size is affected. So you get more drops and bigger drops. So it seems like maybe, there's a lot more things to work on reduced gravity cooking. And the other one would be how far the drops travel, okay? So this is the entire length of our sheet, okay? And we are seeing that, so this is the cumulative number of drops. So like the percentage of drops. And we're seeing that, again, there's like a clear pattern. What happens is, this is the moon one. It seems that they're evenly distributed along the distance. So they don't get, the number of drops don't decrease the farther you go, because this one shows you that it goes up quickly. So almost 1/3 of the way, you get about 80% of the drops, 1/3 of your sheet. But for the reduced gravity, you get more drops farther away. Say, let's say 3/4 of the way, you still have about 20% of the drops are there, as can be seen here too. The blue ones is for the moon. So farther away, there are more drops compared to the one on Earth. So that's something we see. So they travel more, they're bigger, and there's more of them at reduced gravity. This is interesting because, like I said, it's the first time that we've done cooking in reduced gravity, and the data is going to be useful when we make our model. Okay, so that's all I have for now for the data for the reduced part. But we're working on it, and we should be able to get some more. Specifically, I'm more interested in how the numbers come out, I mean the numerical modeling of it. So Bryan, Caldwell, so these are the undergraduates who work with us, looking at all the data, Susana Carranza from Macau, and it was funded by NASA through the Small Business Innovation Research Program in cooperation with Macau and the NASA Reduced Gravity Office, who help us a lot. They're very, very grateful for them because without them, then we would not have pulled it off. Because all the safety procedures they put in, really, I mean, there was no problem. So it was nice. And okay, so that's that. So we were able to do some floating because we were not doing anything during the zero gravity part. So here's us floating around. Nice. Okay. So by the way, if I forgot, I'm from the Philippines, so I put my flag there. Just